The ABO blood typing system is one of the most important ways we have to characterize blood. Like other cells, red blood cells have a variety of proteins on their surface. The proteins that determine ABO blood type are antigens, and proteins are transcribed and translated from genes. In the ABO blood typing system, red blood cells can have A antigens on their surface, in which case the person is said to have type A blood, B antigens on the surface, which is classified as type B, both A and B antigens, which is classified as type AB, or neither A nor B, which is classified as type O. In summary, blood type is determined by antigens. In the ABO system, there are two antigens possible, A and B. Antigens, in turn, are determined by genes. A sequence of DNA can be transcribed and translated to synthesize one of these antigens. Since we have two copies of DNA, one from mom and one from dad, that means we get two chances. If the sequence you got from mom encodes the A antigen and the sequence from dad encodes B, then you will express both A and B antigens on the surface of your red blood cells, and therefore have type AB blood. If, on the other hand, dad's sequence encodes another A, then you will only produce A antigens and therefore have type A blood. The term antigen doesn't just refer to the proteins on the surface of red blood cells. Antigens are anything that can generate an immune response. For example, when a cell is infected with coronavirus, that cell will begin to express a certain antigen on its surface. The immune system can recognize this antigen and generate antibodies to help fight the infection. This antibody can only fight against this specific type of infection. If you get salmonella, the coronavirus antibody can't help you because this new infection will have a new antigen on its surface and therefore requires a new antibody to be able to bind specifically to it. Every time your body is infected with something new, your immune system will generate an antibody that can bind to that specific pathogen. Since there are two possible antigens in the ABO system, that means there's two possible antibodies that the immune system can produce. One antibody that can specifically bind to the A antigen, and one that binds specifically to B. When we collect blood, the sample is referred to as whole blood. But when we spin it down, all the blood cells drop to the bottom of the vial, and all the fluid and solutes rise to the top. This fluid component is called plasma. The majority of blood cells are red blood cells. As we know, A and B antigens are found on the surface of red blood cells. Plasma, on the other hand, is where antibodies are found. The thing to remember about antibodies is that they are made by the immune system to fight foreign antigens. This means a person who has A antigens should not make antibodies against their A antigens, because if a person made both A antigens and anti-A antibodies, then the antibodies would bind to the antigen and trigger an immune response against their own normal, healthy red blood cells. Using this logic, we can deduce which blood types will have which antibodies in their plasma. Blood type describes the red blood cell. Type A indicates A antigens are on the surface of the red blood cell. How do we determine which of the two possible antibodies will be found in the plasma? We don't make antibodies against our own antigens, so there should be no anti-A. But B is a foreign antigen, so this blood type should have anti-B in their plasma. Type B has B antigens on their red blood cells. Since A antigens are foreign to this person, they will make antibodies against A. But B antigens are found on their own cells, so the immune system should not make any antibodies against B. Type AB has both A and B antigens. Since they are both found on their own cells, their immune system should not make antibodies against A or B. Type O has neither A nor B antigens. Therefore, both A and B antigens are foreign to this blood type, and they will make antibodies against them both.